Studying on weekend? So I was talking to a student in the class just before class and was shocked to discover we have an exam on Wednesday. I was shocked. Imagine how you guys felt about it, but it means that it's going to be a busy time because I'm very busy next week, but maybe we should just call it all off. What do you think? You'd like that, yeah. But that would mean we'd have that much more on the final, and the final would be worth that much more as well. What's that? Later in the week. I can't do that, unfortunately. Much as it would be better for me if I could, but I can't. So, open book. Just come in and tell me what you know. There are a couple classes I teach where I give students the option of having an oral exam with me. If they want to come in and I give them an oral exam. I'm too mean, yeah. Actually, several students take me up on it, and they're not that bad. One of the real advantages of oral exams, and when you get into graduate school, you will learn the advantages of oral exams are that once the words are out, they're gone forever. But once you've written them down, they're there forever. So you can say something really stupid in an oral exam, and sometimes people will forget it, you know. But anyway, that's neither here nor there. We have to get through glycolysis and regulation of it today, and that's not that much. So we'll probably get out early today. I know I always promise that, but today I think there's a good chance we will get out early. And before we go to that, I want to announce something for anybody who's interested. In fact, I saw a couple of you in the class walking around campus. I'm making a music video. I've written a new song about OSU. And I'm trying to get scenes of campus activities and so forth. People walking around. You don't have to be an actor. You don't have to do anything. You just walk around. Do something on campus. If you're interested in being in the video, it's going to be a YouTube video. If you're interested in being in the video, send me an email or come see me after class. And we're probably going to do some videotaping on Sunday. So if you're interested in that, let me know. And I would love to have you in it. It should be a lot of fun. And I won't embarrass you. And you won't have to use any lines. It's just campus activities, all it is. Okay? And yes, I will show you the video when it's done, which will hopefully be next week sometime. All right. Let's talk about glycolysis. So last time I took you through the process, and I showed you that there's really, the reason people divide the pathway into two phases is the first phase is the sort of preparation phase. That is, you're getting the molecules ready for what is the energy release phase, which is the second phase. So the first phase takes energy. It requires ATP. All right? The second phase generates ATP. And just as important as generating ATP, I want you to remember that the process generates 2 NADHs and 2 molecules of pyruvate. And those guys have a lot of energy in them that is used to make more ATP. Okay? So glycolysis itself doesn't generate much energy. There's not much energy that comes from the pathway of glycolysis, all right? The energy comes from what the products of glycolysis make, pyruvate and NADH. There we get a lot more energy. Now, that's important to keep in mind, okay? Now, one of the things I skipped over last time were the fates of pyruvate, and that's actually where I'm going to start today, because this is where we begin to see what's happening with energy later, okay? So let's take a look at the fates of pyruvate. So pyruvate, you remember, was the last molecule we made in glycolysis. Okay? It was the last molecule we made in glycolysis. So we've gone from glucose down to pyruvate. We put 2 ATPs in, we got 4 ATPs out, so we had a net gain of 2. We put 2 NADs in and 2 NADHs out, and I sure hate that bouncing thing. Leave me alone, go get a life. Quit. Okay. So we've gotten down to pyruvate. Now, what the cell does next with that pyruvate really depends upon the circumstances that the cell finds itself in. All right? There's 3 possible circumstances, as you can see on here, 2 for any given organism. All right? So one circumstance that's common for virtually all aerobic organisms, that is those that use oxygen, is that for all of them, if there's oxygen present, they can go and donate their pyruvate into ultimately what will become the citric acid cycle. Okay, this is an oxidation process, and we'll talk more about that when we talk about the citric acid cycle, and I'll talk more about that individual reaction later today. But suffice it to say that if there's oxygen present, the middle pathway is chosen by essentially all aerobic organisms. Okay? Well, you say, okay, well, I'm an aerobic organism. That's what my pyruvate is going to do. And the answer is yes and no. All right? So the no part of the answer is that there are times when your cells don't have enough oxygen. Okay? Well, when might that be? Well, it takes something besides putting a plastic bag over your head to have that happen. Okay? That's not the way I recommend having it happen. In fact, you don't last very long if you do that. It happens in your muscle cells when you're exercising heavily. All right? And the reason that it happens in your muscle cells is because your muscle cells can use oxygen faster than your bloodstream can supply it. So when you're exerting yourself fairly heavily, you are causing those cells to be deficient in oxygen, and it's under those circumstances that those cells need to have a separate option. And I'll tell you why that's the case in a second. Okay? All right? So the second option occurs in animals, as you can see on the screen. And it involves taking that pyruvate that was produced and converting it into lactate. Now notice that lactate is different from lactose. Lactose is a sugar. Lactate is a 3-carbon molecule. Lactate. Okay? Now, lactate is reduced compared to pyruvate, and that takes electrons from NADH, and when NADH gives up its electrons and proton, it becomes NAD+. Okay? So in this process, what's happening right here is pyruvate is getting reduced to lactate. Now there's 2 pyruvates produced by glycolysis, so we get 2 lactates, and it requires 2 NADHs. Not coincidentally, up here we produced 2 NADHs. Okay? So the product of glycolysis was 2 NADHs, and down here we take 2 NADHs, and now we convert them back to 2 NADs. And you'll notice these little dotted lines connect things, because if we follow the NADs back up here, they can go back and they can feed this reaction and produce that, and that can feed this reaction, and we've got a nice little cycle. Well, that cycle turns out to be really, really important, and the reason that that cycle is important is because normally when there's oxygen present, NADH that's produced here is dumping its electrons into the electron transport system and becoming NAD. There's no need to have some other molecule to pick them up. All right? So when there's no oxygen, and if we didn't have this pathway, what would happen? Well, if there's no oxygen, we have no way to get rid of NADHs down here. We don't have this pathway. What's going to happen is, once we make NADH, we start running out of NAD. Remember I said we have limiting amounts of NAD and NADH. All right? And when we have limiting amounts of NAD and NADH, then if we start converting NAD to NADH, and we don't convert NADH back to NAD, we're going to soon lose all of our NAD. Well, so what, Kevin? Well, the so what, Kevin, is this. If we lose all of our NAD, we stop glycolysis. Now, that's really a dire situation for a muscle cell, and the reason it's a dire situation is because when there's no oxygen, muscles are relying almost completely on glycolysis as their source of ATP. The cells are frantic at this point to keep glycolysis going so they have a source of ATP. Otherwise, they are hosed. Well, fortunately, our muscle cells do this, and they do this fairly well. And we'll see later there's another cycle that comes into play called the Cori cycle, which I won't tell you about here. But suffice it to say, this enables our muscle cells to keep going. Okay? So, this pathway turns out to be very, very inefficient. We'll see why in a second. It's very, very inefficient, but it turns out to be one of the major ways that muscle cells get ATP when they've run out of oxygen. All right? Well, how inefficient is this pathway? All right, let me give you an idea. If I start with glucose and I get to pyruvate, you've seen that I make two pyruvates, I make 2 NADHs, and I make 2 ATPs. If this pathway goes, the 2 NADHs that I had disappear, and I'm making only lactate here. Lactate is a biological dead end. It doesn't get made into anything else. Okay? So if I have no oxygen, the entire process is giving a grand sum of 2 ATPs per glucose. That's the only things that there of any use to it. Now, on the other hand, when oxygen is present and those pyruvates and those NADHs come down here, what happens is we end up with, for each glucose, 38 ATPs. And people count them in different ways. I count them as 38. If we have 38 ATPs, all right, compared to 2, that means that we get 19 times more energy when we have oxygen. Now, my question to you is, you guys like to exercise. I hope you like to exercise. Exercise is good for you. I'm a jogger. I like to jog. Okay? And when I'm out there jogging, the question is it better for me if I want to lose weight to be aerobic or anaerobic? What do you think? Everybody says aerobic because we've all got this thing planned in our heads that, hey, I want to get aerobic. And the answer is, if you want to lose weight, the best thing that you can get is anaerobic. How come? Right? How come? Well, for every glucose, if I only get 2 ATPs, doesn't that mean I have to burn more glucose to keep myself going? The reason that you lose weight because of exercise isn't because you're going aerobic, folks. It's because you're going anaerobic. Tell that to your exercise instructor. Okay? You're breathing heavily, and people associate that breathing heavily with, I'm going aerobic. And in fact, you are increasing your aerobic metabolism. All right? But the more you exercise, the more you're going anaerobic, and the more you're burning off of energy. Okay? Does that make sense?
Bring it back to class.
What's that?
Bring it back to class.
Bring it back to class. The instructor with the bag over his head, right? So, do you have a question?
Well, I have learned that in aerobic exercise, you'll be burning more fat than carbohydrates.
Yep.
Yeah,
Yep.
Aerobic will burn fat. So, like, for losing weight, aerobic would be the key.
So, her comment is that she's learned that when you're exercising, you're burning more fat than you are glucose, and so the key is being more aerobic to burn more glucose. Okay? The answer is they both require oxygen. They both require oxygen. All right? So any process that less efficiently uses materials is actually going to be a better thing for weight loss. The less efficiently you use something, the more weight you're going to lose. So that's true whether it's fat or it's glucose. Yeah. Nobody believes me now, but it's true. Well, if that were all we had, it would be 19 times better. Now, I'm exaggerating the situation a little bit in the sense that you're never really completely out of oxygen. Okay? So it's probably somewhere in between there. But nonetheless, when you're going anaerobic, you are, in fact, using energy, using your resources a lot less efficiently. Now, as we will see later, there's another cycle that kicks in called the Cori cycle. And the Cori cycle is really a cool cycle, but we're not far enough along for you to understand the Cori cycle. The Cori cycle is there to feed your muscles. It's there to feed your muscles, and it comes from your liver into your bloodstream, and it's a cool process as well. All right. Well, speaking of cool processes, there's even more that's cool with respect to pyruvate. Okay? The third process that's up on the screen that I haven't talked about is this guy right over here. All right? Under anaerobic conditions, that is when they run out of oxygen, bacteria and yeast, what do? Well, they convert pyruvate into ethanol. That's the process of fermentation. Now, people think of the term fermentation, and the first thing that comes into their head is beer, wine, ethanol, alcohol, right? This is fermentation too. All right? Making lactate is fermentation. Any kind of anaerobic metabolism of pyruvate is fermentation. Okay? So just because you think fermentation, you think beer and so on and so forth, that's not exclusively the way the term is used. Fermentation includes the metabolism of pyruvate to make lactate. Okay. Now, notice that what yeast and bacteria are doing is exactly the same thing that we're doing. They're converting NADH back to NAD so it can go back and keep glycolysis going. They're keeping glycolysis going. Just for the same reason that we're doing it, we're keeping glycolysis going. All right? In the case of the bacteria and yeast, all right, they basically spit this out and keep that going up here. In this case, we get a chance to recover. If we went anaerobic for too long, we wouldn't be alive, as you well know. These guys can go quite a while without that and still produce, still grow and produce things. Okay, questions about that? Okay. Let's talk a little bit about, let's come back down here and talk about, well, first of all, there's this reaction. That's the reaction I was talking about, pyruvate going to lactate. There's an enzyme. You don't need to know the enzyme name, okay? But you can see what's happening. This ketone group is becoming an alcohol. So we're reducing. That involves a reduction, requires electrons from NADH and protons as well. And we come up here and we've got that. Okay. Now, the more interesting one is actually the one involved in alcoholic fermentation. All right? And that's this guy right here. So in alcoholic fermentation, it's actually a two-step process. So in the first step of the process, pyruvate is converted by a decarboxylation reaction, that means a carboxyl group is lost, into acetaldehyde. Now, acetaldehyde is something that if you don't know, you probably should. Okay? Anybody know why you should know what acetaldehyde is? One word. What? Hangover. Okay? There's your hangover molecule right there. Hangover. All right. Now, in bacteria and yeast, they take that acetaldehyde and they convert it into ethanol, and you drink the fruits of their labors. Right? Now, the interesting thing here is this enzyme, alcohol dehydrogenase. Alcohol dehydrogenase converts acetaldehyde into ethanol. And interestingly enough, human beings have the enzyme alcohol dehydrogenase. We have plenty of alcohol dehydrogenase. Why do I have to spend so much money on beer? Which I don't, but I mean, let's imagine I did, okay? Why do I have to spend so much money on beer? Can't I just make my own? Well, the answer is no, we can't, because we can't go through this reaction from pyruvate to acetaldehyde. So then the question is, well, why in the hell do we have it? Why do we have this enzyme to do this reaction? What's going on? If I didn't have that enzyme, I wouldn't have a hangover. Any thoughts? There are bacteria in our guts, but that's not the source of what's going on. But it's good try. You're doing the reaction backwards. That's exactly what it is. Okay? So the body is detoxifying ethanol and making acetaldehyde. So in your liver, that's what's happening. It's running the reaction backwards, and acetaldehyde can be readily oxidized to make acetic acid. Some people say it's the acetic acid that gives the hangover. There's actually argument over that. All right? But either acetic acid or acetaldehyde, one of them gives you the hangover. The point is that the body is trying to detoxify ethanol, and that's why it has alcohol dehydrogenase. Another thing you probably don't know is that virtually everybody has blood alcohol. Did you know that? Even if you don't drink. You have alcohol in your blood. Tell that to the officer, right? Okay? The reason you have alcohol in your blood is because of those bacteria. So you've got bacteria deep in your gut that are actually making a very tiny amount of alcohol that gets into your bloodstream. So everybody has a baseline level of alcohol, even people who don't drink. Okay. All righty. Let's see. What else here? Don't need that. We'll talk about that later. And we've got this guy. Okay. What do I want to say? Okay, actually, let me back up. All right. Now I'm going to talk about this reaction here more later. When we talk about This reaction actually does occur in us, except for we don't stop at acetaldehyde. We make acetyl-CoA. You've heard of acetyl-CoA. So we have another step that we add to this process, and I'll explain more about that later. But I'm showing you this enzyme because it's an important user of vitamins and vitamin-like things. Okay? This enzyme is called pyruvate decarboxylase, and pyruvate decarboxylase takes that carbon dioxide off of pyruvate and makes CO2 and leaves you with acetaldehyde. That's what this reaction is producing. This reaction requires several coenzymes to occur. Well, one of those coenzymes is thiamine pyrophosphate. That's why we have to have thiamine in our diet. Another thing that's required for this reaction, for this overall reaction, is lipoic acid. It's also a vitamin-like substance. Okay? In addition, because there's an oxidation that actually happens later, we have to have both NAD and FAD. There's 4 things. And the fifth thing, there are 5 things this reaction has to have. The fifth thing that is needed in this reaction is coenzyme A. And yes, I'll repeat these, okay? Coenzyme A is what the acetyl group gets put onto to make acetyl-CoA. All right, so this enzyme is a pretty big user of vitamin-like things. They include thiamine pyrophosphate. You can call it TPP if you want to. Lipoic acid, LIPOIC. NAD, FAD, and coenzyme A, or you can just call it CoA if you want to. All right, so we need vitamins to make NAD and FAD, 2 other vitamins, lipoic acid and thiamine pyrophosphate. This reaction takes a lot. It turns out this enzyme is almost identical to another enzyme that's in our citric acid cycle, and they both use the same things. They both have a very similar enzyme reaction and a very similar mechanism, and they both use the same things. And I'll say more about those as we go forward. Okay. Questions about that? All righty. You guys aren't very talkative today. This is reminding us what's happening in that reaction to make lactic acid. Okay? In this reaction, we are keeping the glyceraldehyde 3-phosphate dehydrogenase reaction going. That's the one oxidation reaction in glycolysis. And that one oxidation reaction in glycolysis is necessary because otherwise if we stop this, then glycolysis can't keep going. Okay. The same thing is true for yeast and bacteria as well. You might wonder about plants, but plants don't really go anaerobic. Plants don't exercise. Okay? And so they don't really have those conditions where they're going very anaerobic. All right. The last thing I'm going to talk about today, and there's magic words for anybody, is I'm going to talk about this. I'm going to talk about regulation. All right. So regulation of this pathway is very, very important. All right. And you can see it's a little complicated. As I said, this is an unusual pathway in that there are 3 different places where the reaction is regulated. And the regulation of the pathway does not occur, one of the regulation steps occurs at the very end. All right? So I need to tell you a little bit about this. Let's talk about the 3 enzymes first. So the first enzyme is hexokinase. And its regulation is complicated enough, I'm not going to say much about it. I just want you to know that it's one of the regulation enzymes. Okay? The second regulation enzyme is phosphofructokinase, otherwise known as PFK. That's the enzyme that catalyzed reaction number 3. And that's, as I said, the most important and the most interesting one that's there. The last enzyme that is regulating glycolysis is the last enzyme of the pathway, and it is pyruvate kinase. Pyruvate kinase. Now, let's talk about how these are regulated. So, first of all, let's talk about PFK. PFK is regulated in a very interesting way. All right? You'll notice it's activated by AMP, And AMP we've sort of talked about in the class. What's ATP? ATP is high energy, right? We break down ATP, what do we get? ADP, right? And if we break down ADP, what do we get? So when we break down ADP, we get AMP. When cells are running out of ATP, they start breaking down ADP and making AMP. So a cell that has much in the way of AMP is going to be very much in need of energy. Very much in need of energy. Okay? We can think of it as being in a low energy state. Well, it turns out that being in a low energy state activates the enzyme. That's pretty cool. Okay? Because why? Well, this pathway, as you can see, what does it do? It produces ATP. And it produces other things that can be used to make ATP. So this pathway gets turned on by conditions of low energy. Cells have amazing checks and balances in them to make sure things are made in the proper amounts. Okay? Now, AMP is an important regulator, but there's a more important regulator of this enzyme. And it wasn't even discovered until about the 1980s. And it's this molecule here that always confuses students. It's called F26BP. And the reason it confuses students is, well, it sounds an awful lot like F16BP. It's not the same. Right? And second, they say, well, where is it made in the pathway? And the answer is, it's not. It's made somewhere else. Why they don't ask the same question about AMP, I don't know, because it's not made in the pathway. But I always get this question, where do we make the F26BP on here? Well, it's not made here. It's made somewhere else. All right. Now, F26BP, the reason it was so hard to discover was it is so effective in turning the enzyme on. It takes very tiny concentrations of it, and there's so little of it in the cell that people didn't really realize it was there or that it was important for a long time. This is a very potent activator of phosphofructokinase. Now, what's not on the screen, well, actually it is on the screen, I'm sorry, it's right there. What also is on the screen, that's important, is the fact that the enzyme is inhibited by ATP. The enzyme is inhibited by ATP. What does ATP, high concentrations of ATP, indicate to a cell? High energy, right? And when I have high energy, do I want to be burning my glucose? Well, no more than I want to be running my furnace in July. I don't want to be burning my glucose when I have high energy. And so ATP concentration goes up, PFK gets inhibited, the enzyme, and you stop any one of these enzymes, and you're going to stop the reaction. Or stop the entire pathway. I'm sorry, stop the entire pathway. All right. Now, the other enzyme that's interesting is this guy down here. Okay? One of the activators is missing. I talked about it last time. But this enzyme, pyruvate kinase, is actually activated by 3 different things. Okay? It's not F26BP. That's a mistake. Sorry about that. It's activated by AMP. It's also activated by this guy up here, fructose 1,6-bisphosphate. That should be a 1 instead of a 2. Okay? This F16BP comes from up here. That was that feed-forward activation that I talked about last time. And feed-forward activation helps this process to go forward. Now, I'll tell you why in a second we regulate this last step in the pathway. But before I do that, I want to remind you that this guy is inhibited by ATP for the same reason that this guy is inhibited by ATP. We don't want this process going if we have plenty of energy. All right? Now, let's think about this reaction right here. What did I call this reaction when I described it to you the other day? I said this was a big something. Big bang, right? This was the Big Bang right here. And the reason it was the Big Bang was it was energetically very favorable in the forward reaction. It was very, very favorable in the forward reaction. Okay? Now, why does that matter? Well, it matters for the following reason. All right? On Monday, I'm going to talk about a pathway called gluconeogenesis. And it's how we make glucose. Our body makes glucose just like it burns down glucose. And it uses most of these reactions in reverse. The ones it doesn't use in reverse are those that have very high energy. Here's one, and here's one. The three regulated reactions of glycolysis are bypassed in gluconeogenesis. We'll talk more about that later. But more importantly, this guy takes a little two-step. It makes something called oxaloacetate, and then oxaloacetate gets converted into phosphoenol pyruvate, and then everything starts reversing. That is, everything starts reversing so long as it doesn't get converted back into pyruvate. Remember, this is a very energetically favorable reaction. Let's imagine I'm a cell. I'm trying to make glucose. The conditions under which I'm going to make glucose are going to be what? What state is my cell going to be in when I'm making glucose? High energy or low energy? High. Okay? Because it takes energy to make things. So when my cell is full of energy, it says, I better store this as something, and so it makes glucose. All right? Well, if it wastes energy making oxaloacetate and then making phosphoenol pyruvate, and then this pyruvate kinase is still active, what would happen? Bang. It would not go up and make glucose. It would just come back down and be pyruvate again. So regulating this last step is done to enable the cell, when it needs to make glucose. Okay? So that last regulation step is really important because that reaction here is so energetically favorable. Very, very important. Now, that's a lot of material. Thoughts or questions? Yes? What's that? I think I said pyruvate kinase is activated by 3, but I took it back. There actually is a third, but it's not on there. There's an amino acid that affects it also. Yeah. But that's okay. I'm not going to put it on there and add it to your burden of memory. Yes? Down here, this should be a 1 down here. Up here, it should be a 2. Okay? So the one on the bottom is a 1. The one on the top should be a 2. The hexokinase, like I said, it's complicated enough. I'm not going to ask you to know that. Just know that it is a regulatory enzyme. Okay. Well, I have a bit of a surprise for you today. And the surprise that I have for you is I'm not going to sing a song, but I actually have a music video that I've done previously that I think you will enjoy. And it's relevant to our weather outside. Okay? It is called, if I can find it. Here we go. Whoop, what happened here? All right. See you guys on Monday.